hopefully you're adequately caffeinated, and so I won't put you to sleep. So we were talking about the immune response in model systems, mostly to sandfly saliva and Leishmania infection. So now we're going to switch a little bit and describe some of the studies that have been done in humans as we're kind of moving towards this idea of using saliva as a vaccine. So this group started to ask this particular question in humans instead of mice about the type of immune response to Lutzomyelongiopalpus. And so they recruited, and this is done in Brazil, and they recruited humans and exposed them to the bites of Lutzomyelongiopalpus. Each one of these dots is an individual. And if you look at days post sandfly bite, and they're measuring in the peripheral blood this cytokine or this protein, interferon gamma, which remember is the protein that's indicative of a Th1 cellular immune response. And so this is the kind of response that we want to induce, because that induces resistance or killing of Leishmania parasites. So they test that, so they expose the individuals on day 0, and so this is the background level that they see. After 60 days or 2 months, they test again, and they see a pretty good response of interferon gamma. So in the humans, we're getting the response to the sandfly bites that we would want, number one. Number two, it's not surprising, because remember I showed you those awful sandfly bites, and so it was already, we thought, would probably do that. The interesting part here is they wait till after a whole year of exposure, and then they expose them one more time, and they see that there's a memory response after 1 year of exposure, showing that this exposure is long-lasting, which is important. Oops. What did I say there? I don't know what I said. Oh, so we've switched from our model systems now to studying humans, but of course this isn't natural exposure. This is they're coming into the lab and sticking their arm, I suppose, in the insect cage or something like this. So the Valenzuela group decided to test in a natural setting, but they did also expose the individuals, and in this case they're looking at the immune response to Phlebotomus papatasi, because we are in Mali, we're in the new or the old world, and in that area Leishmania major is transmitted endemically by Phlebotomus dubosci. And so in this case they're looking at the peripheral blood mononuclear cells from individuals that have been exposed to Phlebotomus dubosci. And they're looking at these cytokines or these proteins that are indicative of a cellular response, interferon gamma, as well as interleukin 12. I haven't talked about this one, but it's also a protein that's associated. They also looked at these proteins, interleukin 10, 13, and 5. These are indicative of a Th2 immune response, which is the opposite of a Th1. So remember I talked about this is associated with cellular immunity. This is associated with antibodies or humoral immunity. So each one of these is an individual, actually the same individuals, and they're looking to see what their response is to Phlebotomus papatasi bite, or dubosci bites. And you can see it's somewhat mixed, right? I think what we were hoping is we would see this high and these really low, but it seems to be somewhat of a mixed response. Interestingly, it looks like you have a subset that produced very high interferon gamma and a subset that produced low. And that is very similar to what they saw in the non-human primates. So they wanted to look at this and break this down a little bit more. Remember the non-human primate study where they looked at the high interferon gamma producers as opposed to the low interferon gamma producers, and they saw a difference in the resistance and the antibody responses? they wanted to do something similar here. They're not challenging these individuals with parasites. They're just looking to see how they are influenced by bite. So what they did was they took these individuals here and looked to see what other cytokines they were producing. And then they took these individuals to see what cytokines they were producing. And so they were able really to break it down into 3 groups. So those that produced high amounts of interferon gamma, they saw that a lot of them produced lower levels of these Th2 cytokines, interleukin 3 and interleukin 5. Those individuals that produced lower interferon gamma had higher Th2 cytokines. So those dotted lines are indicating an individual person, right? So this is the interferon gamma production of this person. Down here is their IL-4, down here is their IL-5. So in the response to these sandfly bites, they have people who respond in a Th1 response, like cellular immunity, those that produce a Th2 response, and those that just are kind of mixed. So if we were going to use this as a sort of vaccine, or even if you're just trying to study epidemiologically how people would be affected by uninfected sandfly bites, it looks like you have different responses. That's not surprising, right? These individuals all have different histories, different genetic makeup, probably different diets, different infection, been exposed to sandflies differently. So all of that is really not that surprising, but it is indicative of what happens in nature. And if you're trying to make any vaccine, and not just with saliva, sandfly saliva, to anything, you're going to have this diversity of responses. So the interesting other part that they did in this particular study is they took individuals of different ages and then they exposed them to sandfly bites to see how many of those individuals reacted to the sandfly bite, so got at one of these DTH responses. So we didn't know that much about sandfly bites, about this idea before, but certainly with Anopheles bites for malaria, we know that at least in Africa, I don't know about other places of the world, people become tolerant to the bite of the sandfly bites. So you know you can have individuals who, we get our normal mosquito bite, you itch it and then it goes away. We have some people who are allergic, right? They get big welts and big red marks. And then we have people who don't respond at all, so that they kind of become tolerant. And what's thought is like over time that you're exposed to these bites over and over and over and over and over, and eventually your immune response thinks it's normal and doesn't respond anymore. In some places in Africa, you can see people sleeping at night and they might have 1,000 mosquitoes on them, and they don't even notice. They don't feel it, they don't get a swelling, and that's called tolerance. So if you were going to use a salivary protein in a vaccine, and the person is tolerant to that, they're not going to have an immune response, it won't work. And so that's what they're testing here. And so they did find that some of the older individuals seem to be less reactive, although pretty good age division is still reactive. So it's possible that you can get tolerant to sandfly bites, although it's much later. So this idea of tolerance, you need to understand what you would be, what's happening in nature to see if your vaccine would be effective. So interesting, so these studies that I showed back here were done with peripheral blood mononuclear cells. So that's systemic response. But maybe more importantly, what's happening at the bite site, right? Because if you're challenged naturally, you will be challenged with in a site with saliva and the parasite. So they wanted to see what was happening actually in the skin where the parasites bite. So they took biopsies of where, sorry, not parasites, sandflies, where the sandflies were biting, and then they measured mRNA of various cytokines at that bite site. And they found that they saw interferon gamma that was upregulated, which is what we want. So that looks great. But what's in the materials and methods that you might not notice is they only looked at those individuals that were Th1 responders. So they didn't look at all of the individuals. So they biased their results by only looking at those that they thought would give them a good response. But this brings this idea of immune correlates. Either here in that PBMC study or here could be an immune correlate. So when you have vaccine discovery and you think you have a vaccine, so I talked about how you start in mouse models and then you go to non-human primates and eventually you go into humans. But before you can do a phase 2 or phase 3 clinical trial where you're testing for efficacy to see if it protects against your disease, you first test to see if it will give you the response that you want. And so in that context, interferon gamma would be the immune correlate. So you would take a bunch of people and you just vaccinate them to see if you induce interferon gamma, and that's your immune correlate. And so that's part of this process. So in that context, those individuals, even though they were living in Mali where they could be naturally exposed to Phlebotomus dubosci bites, they were also given bites of Phlebotomus dubosci to see what would happen with their response. These individuals in Tunisia were studying the response, the natural response to Phlebotomus papatasi. So we've gone from Mali, where Leishmania major is transmitted by Phlebotomus dubosci, to Tunisia, I guess I'm going the opposite way, to Tunisia, where it's transmitted by Phlebotomus papatasi. So in this case, they took individuals that were living in an endemic region and naturally exposed to Phlebotomus papatasi bites, and people from a control region that would not be exposed to Phlebotomus papatasi bites. They isolate their peripheral blood mononuclear cells, and they look to see if those cells will proliferate, they'll divide in response to salivary gland homogeneity, essentially. And so part of the immune activation, the first thing that happens is those cells that get activated and they divide. And then they produce their protein or their cytokine. And so the first thing you'll notice is, seems like there's very few individuals that are proliferating, even though we know they're being exposed. I mean, not much different than compared to the control region. So it doesn't look as if their cells are being activated. And so then they also looked to see about the interferon gamma production of these individuals. And even if they are the ones that did proliferate, they didn't really see any interferon gamma production, whether they proliferated or not. And then the interesting part is they looked at one of those Th2 cytokines, if you will. They saw that those that were proliferating were actually producing large amounts of that inhibitory cytokine. And so the opposite. So this is what's happening naturally, but it seems to be opposite of what was predicted. So we know, immunologists know that this cytokine actually inhibit interferon gamma. So they wanted to test to see if that's what was actually happening. So they took And they took these individuals not the individuals, their cells in culture, and they treated it with an antibody. So here's salivary gland extract, which is the same as homogenate. And they put anti-IL-10 to see if they could reduce or switch the proliferation response. So they found out if they block IL-10, they get more proliferation and more interferon gamma production. So they further went on to show that the interleukin-10 is coming from a subset of T cells, CD8, and the interferon gamma was coming from a subset of T cells called CD4. Has anyone heard of CD4 and CD8 before? In what context? Where did you hear CD8 and CD4 before? HIV/AIDS, right? People who, so HIV infects CD4 positive cells, and when they present with AIDS infection, it's when the ratio of these two cell types changes. So these are important cells in the immune response. And we know that the interferon gamma is coming from CD4 cells. So obviously people who are HIV infected are going to be more susceptible to leishmaniasis. Okay, so we've gone from these model systems in the laboratory, right, studying these flies that are in colony. It works really well in inbred strains of mice, this resistance, this inducing resistance. Works okay in non-human primates, maybe not as good as we'd want. It seems like we're inducing the right immune response in humans. But Paulo Pimenta, who is also from Brazil, started asking these questions about, well, most of these experiments have been done with colony flies, right? So you're growing sand flies in the insectary in the laboratory, breeding them. They tend to be inbred. They're all feeding on the blood that you're either buying for them or from mice in the laboratory. Likely any bacteria or microbiome that's associated with them are the same. So it's all very controlled. So he started asking the question about, well, what's actually happening with the sandflies in the wild? What's happening in the field? And so he's using that same model that I showed you before, but he's actually just testing this part. Would the saliva from wild-caught flies give you this response? So here he has parasites alone, and there's your lesion size. And he's injected them with colony salivary gland homogenates from colony flies. And this is Lutzmia longipalpus. And now he has switched to a different parasite, Leishmania amazonensis. And he sees the exacerbation effect that we've shown you before. But now when he uses the salivary glands from flies that he went out and collected, he sees that that's not statistically significant, suggesting or indicative that maybe there's something funny about what's going on with these colony flies. Or maybe not. Maybe just the wild-caught flies have different bacteria associated with them, and it's that bacteria that might be influencing the response in some way. As we know, it turns out the microbiome does everything, right? It messes with our metabolism. It changes all sorts of things. So it could be the same for the immune response, and it can be the same for the sandflies. Elias Zouhaoui in Tunisia asked this question in terms of the resistance phenotype, right? So if you're vaccinating with salivary gland homogenate, will it change the response? And he's working with Phlebotomus papatazi because he's from Tunisia, and that's where Phlebotomus papatazi is endemic. So again, he's looking at lesion size. He actually doesn't inject into the ear, he injects into the foot pad, but it's the same. Where he's vaccinating 3 times with colony flies or wild-caught flies, and then challenging with Leishmania major in the presence of saliva. So in yellow, whoa, you can see what happens if you just inject with sandfly saliva. If you vaccinate first with sandfly saliva from colony flies, you see the resistance phenotype, so the controls work fine. But if he does the same experiment with wild-caught flies, it doesn't seem to work. So Paulo Pimenta showed that the exacerbation thing is not working, and Zuhawi showed that the resistance isn't working. So again, it could be these wild-caught flies have different bacteria associated with them, and it's the immune response to the bacteria, maybe. Or it could be the genetics. So if you see this F39 here, that means that that colony has been inbred at least 39 times. Right, so if you take 2 flies and you breed them, the progeny is called the F1, right? And then if you take those siblings, those F1s, and you breed them, it's called an F2. And then F3, F4, F5. So this means that this has been inbred 39 times. So he decided to test that. Actually, I think some reviewers, when he tried to publish this, made him test that. That what happens when you take sandflies from the wild and inbreed them over time, what happens to the response? Oh. And that's here. So he took F2s, F1s, and F4s, so only a few generations of wild-caught flies in colony, and he did not see the resistance to be induced. It took at least 5 generations for that to happen. So that could mean that this colony from those wild-caught flies is becoming genetically more inbred, and whatever that genetic phenotype is leading to this response. But it could also mean that the microbiome is changing. That whatever the bacteria that survive in colony are different than those that come from the wild. And so that was not controlled for at that time. So it could be microbiome or genetic diversity. So remember I talked a little bit about the timing of vaccination, how you have to work out how many times you give a vaccine, how many times you get boosted. Is it DNA vaccination or is it recombinant protein? Oh, so Petrovuscru and Eva decided to test this in the mouse system, same mouse system that we're showing over and over. So they have different scenarios. They have the control, of course, which is no vaccination. They vaccinate twice or 3 times, it's not showing here. Just prior, like 1 week, 1 week, or 2 weeks, 2 weeks, just prior to challenge, which is the standard that I've shown you on almost all the studies that I've shown you. It's a vaccine, vaccine, vaccine challenge in a short period of time. They also tried to mimic what would be happening in nature, right? People are probably being bitten every day. So what's happening if you're being constantly exposed to sandfly bites prior to challenge? They looked if it was a long time ago that you were exposed. So maybe somebody who visited an endemic region was exposed, then went home for a while and then came back. And then they mimicked maybe an endemic person who moved away and came back, who had been exposed a lot, moved away and then came back and was exposed to infection. So what did they find? So in black is the control. Right, so here's the control. Regular lesion if you're just infected in the presence of saliva. There we go. The normal response, exactly what we'd expect. Okay, maybe I should just give up on the animation. All right. So in all of these other scenarios, which wasn't the standard way, they saw that, in fact, the lesions were still large. And so the timing that you use this vaccine will clearly make a difference. And so perhaps that model system is somewhat skewed in what you would actually see in a natural situation. So timing is important. Okay, so we've talked about all of the diversity of things you have to think about. I mean, it's certainly important for saliva, using saliva as a vaccine, and more complicated if you have a host, a pathogen, and a vector. There's a lot more of diversity of interactions that you have to think about, but all of the things that I presented would be important for vaccine discovery if you're just doing the pathogen without even the vector. Certainly it's increased with the vector complication, but it's the same. So we talked a little bit, and I presented a little bit about the wild-caught flies and some of the ideas about what the diversity might be, right? We talked about the genetic diversity. We talked about it could be microbiome, for example. So this is now some work that we've done where we're trying to assess what is the genetic diversity of these sandflies in nature. So you could imagine if you have two towns, if you will, site 1 or town 2, that are exactly the same. The humans are genetically the same. Let's say they have the same Leishmaniasis species. They only really have one species of sandfly that's biting them. And the thing that I didn't tell you is that sandflies mostly feed on animals. And then they only, in some places, if the only animal available is humans, they're going to feed on the humans. But generally, they're feeding on other animals, rodents, dogs, donkeys, what have you, chicken. So let's imagine that these two villages are the same. Sandflies don't fly very far. They tend to not go very far. I don't know how far do they fly. He's the entomologist. He knows more about this than I'm losing the right thing. So it could be that these flies are far enough apart that they're not breeding. But we don't understand that over time, maybe we'll have some populations and there won't be any change. So we started studying this, and we did at least a study on a lot of red and blue are just structures. But we, or others and us, have found various sites among amino acid sequences that are variable. If we look at different flies, these residues are variable. There's lots of variability out there. If we predict the T cell activation that went on, so remember we were talking about the immune response is cellular. It requires T cells, CD4 positive T cells, and if we predict the epitopes or the portions of the protein stimulated, many of them are in these variable regions. But there are places that aren't. So perhaps, in fact, there is some conservation. If we look at the genetic difference of this protein between these populations, these are the Jordanian ones. Here's one on Molecule. If you compare the two Jordanians, numbers quite low, which is indicative that these are probably fashion that they do. So what happens with the immune system? So I've shown you all this diversity, but does it really matter? So we are able to assess what proteins humans are responding to in response to sunlight because of the work of Jesus Valenzuela, where he characterized all of those particular proteins. Because of their size, we know what ones they're responding to. So we've taken individuals from these sites. Here's a control person in this study. We've isolated the blood from people who live in some of those endemic regions that I showed you. And we've done this in the Middle East for the sites that I showed you. And we've also done this for U.S. soldiers that have gone to Iraq. People endemically exposed, as well as people who are exposed by travel. And so if we look at the endemic sites, their antibodies, this is antibody measurements. We don't know what their cellular status is. These guys are, looks like they've been exposed to antibiotics. Our control people are not exposed, so that looks good. Then if we happen to look at a different group of individuals, again the exposed ones are responding, but those that are unexposed are actually reacting to this antibody. So what could this be? It could be that these individuals actually forgot that they were bitten by sandflies, that they actually did go somewhere, they didn't know that they were bitten by sandflies. Maybe that our control region is not a great one. And it turns out Amman in Jordan is not a great control because Jordan is so small and people travel so much. Cairo is pretty good. This is actually from U.S. soldiers that went to Iraq and those that didn't go to Iraq. So what could this other mean is it could mean that there's a cross-reaction. So that these individuals weren't exposed to Phlebotomus paucatazi, but they were exposed to some other sandflies. Maybe these guys actually were traveling in Brazil, or maybe they were exposed to the bites of lots of mosquitoes. And even though there is diversity in the salivary proteins of all these blood-feeding arthropods, there is some conservation. And so maybe they're getting a cross-reactive response. So why is this important? If you use a protein as a vaccine and it works great, it causes them to be resistant to Leishmania infection and Leishmania lesions. That's fabulous. But maybe there's also a cross-reaction that could cause some complication for some other vector-transmitted disease, so some unintended consequences. So these are all things that we need to know and understand. So cross-reactivity. Oh, lordy, lordy. Okay, everything that I've shown you and the vast majority of studies assessing assessing saliva as a vaccine have been done looking at leishmaniasis, probably because that was the first one to look. A lot of people have assessed vector-transmitted diseases and the role of saliva for viral infection, which is, hot topic now, right, because of Zika. So most of those studies have not studied or not assessed pre-exposure to uninfected bites, but they have studied this enhancement effect. And they certainly see that maybe the saliva is enhancing the response. Those that have studied the pre-exposure have found that it doesn't cause resistance. If it does anything, it makes it worse. More like that intermedia brasiliensis thing that I showed you. So that would be the dogma, at least, and maybe in various situations it will be different. We tested this in that mouse model of a mouse model of infection to malaria infection. And, of course, there we go. So what we did is we pre-exposed Valb C mice to the bites of Anopheles Stephen's eye, so Anopheles mosquitoes, and then challenged them in the presence of Plasmodium uelii, which is a rodent malaria, and Anopheles saliva. And we found that we saw a reduction in the parasite burden in the pre-sensitized individuals. So again, we saw that this pre-exposure could influence malaria infection. Note, just like the Leishmania, this is not sterile immunity. We can't use sandfly saliva, well maybe I didn't test sandfly saliva, mosquito saliva as a vaccine by itself to protect against malaria infection. But you can't seem to do that for Leishmania either. So it's not sterile immunity, but it does seem to influence the response. When we look at the bite site to see what kind of response it is actually a type 1 response, interferon gamma, just like it was for the Leishmania sandfly situation. We also know interferon gamma is important for resistance to malaria infection, at least the first part of the malaria infection. We tested this in a different mouse strain, and it doesn't work. So we don't get the same cytokine response. We didn't test the challenge, but we found that we couldn't induce more interferon gamma, so it didn't work in a different mouse strain. But we did show in the one that it worked that interferon gamma is essential. So here's what I basically showed you before. The resistance with presensitization in wild-type Balb C mice. When we took mice that did not have interferon gamma, so we knocked out, well, we didn't, we bought them, but where the gene